Okay, well, thanks very much for having me here. It's really a pleasure to be able to talk to you about some of the things we're doing and hopefully convey some of the excitement of the field that we're in. How many of you know something about biology? That's pretty good. Close enough. All right, then hopefully you'll be in there for today's talk. So really what we're going to be ultimately talking about is one's health. And I didn't bring a pointer. I don't know. Is there one? Right up there, otherwise I can probably use, there we go, use the arrow here. So really your health state, if you will, is a product of both your DNA and I'll use the term your genome, which is all of your DNA in a given cell, so it's your collection of bases, which encodes all of your genes, and we have about 20,000 genes. And so it's a combination of your genes, if you will, and your environmental exposure. And your environmental exposure can mean the food you've eaten, the pathogens you've been exposed to, the toxins that you may have come in encounter with over the years and such. And so really the combination of these will essentially affect your health. And really, if you ask me, a goal of us in science and medicine is to really understand this equation so well that we could be predictive from a DNA sequence as well as any environmental exposure as to what sort of outcome you might expect for a health state. It's almost the Gattaca, if you will, for those who have seen that movie or familiar with this. So what I want to do is tell you a little bit about this field, and in particular, it's really the first part. We'll spend a lot of time. These days, it's now possible to understand a genome in incredible detail, okay, because there's been a revolution in DNA sequencing. And so what I'm going to do is tell you about a little bit of background about DNA and genomes and our genes, how we sequence DNA, and then with that in mind, I want to launch into the field of personalized medicine, and then I'll tell you a very personal story about me from sequencing my genome, how it led to my discovery that actually I was at risk for and then got diabetes and what was done about it. So this, I think, is a glimpse of what's to come for medicine in the future. So by way of background, I think all of you know we all start out as a single cell. Sperm fertilizes an egg, and we go from this one cell state, if you will, to something that has 5 to 100 trillion cells, so lots of cells, okay? And so it's our genetic program that takes us from here to here, but a classic case of environmental exposure, steroids probably played a role somewhere in this pathway as well. So the combination of these things leads to this final state, if you will. Now our DNA, many of you know, has 6 billion letters, if you will, or 6 billion bases is there. There are 4 chemicals that are strung together in a row, and we have 6 billion of these that makes this code, if you will, that gives us the instructions for how you get from that single cell to the 100 trillion cells. Okay, and 3 billion of these come from our father and 3 billion from our mother. And basically, they're packaged in such a way that we have a total of 46 chromosomes, 23 from our father, 23 from our mother. And again, together, this actually constitutes our DNA. That's something what the chromosomes look like when they're ready to divide, cells are ready to divide. And again, this would be the set, and that would be our genome, if you will, the collection of DNA that's in a single cell that programs us to form a human being. Now, we're all not identical. You look around at one another, and we're not identical for several reasons. One is our DNA is different from one another. So you differ from the person sitting next to you by DNA base changes. And so there'll be single base changes in the 6 billion bases that will make one person this way and another person that way. And then there are also modifications to the DNA. It's called epigenetics or epigenomics that can affect this as well. In fact, there's a total, it turns out, of one change every, single base changes, as they're called, single letter changes, every 1,200 bases or so. So 1 in 1,000 bases will differ, again, between you and the person sitting next to you. And so when you average over the genome, there's a reason, this isn't exactly 6 million, but it's 3.5 million differences between you and the person sitting next to you, okay? And so this happens to be Yao Ming, the famous basketball player, and some guy named Steve. I downloaded this from the internet, and you could see they differ by that many. But they not only differ by these single base changes, they'll also differ by something larger DNA changes that are called structural variants. Amazingly, there are big chunks of DNA that will differ between individuals. These are called structural variants. if this is a reference genome, if you will, and I'll come to this in a minute, you can get deletions. Sorry, this is a bit spotty here. You can get deletions or duplications or insertions. And you actually have inversions, rearrangements. And there are a lot of these. There's 1,000 of them, again, that differ between you and the person sitting next to you. And this leads to lots of changes in our genome that makes us different from one another, okay? So these are the variants we have. They're single base variants of these large structural variants. And some of these, by the way, can be a mega base, 1 million bases that will differ, again, between you and the person sitting next to you. It's amazing how big some of these changes are. So many of you may know there was something called the Human Genome Project. It was actually launched in 1985 to sequence the DNA of an individual. And they didn't do it from one individual. They took a pool of 15 people, although in the end it's mostly 5-ish people, whose DNA were thrown into this pool and then sequenced to develop a reference genome, if you will, essentially decode the string of letters for those 6 billion bases. So put them in the right order and understand what they were. And this was a huge project. Again, it meant determining the order of these 6 billion bases. Thanks. Round 2. Yes, much better. Okay, and this is a massive project. It involved 2,000 people. The cost is still debatable, but it's somewhere between $500 million to $1 billion. Okay, there's a draft finish in 2001 and a more completed version, if you will, in 2003. Don't let that fool you. There's still a few gaps in the human genome we haven't filled in yet. There's still some holes there, if you will. And there were machines. This was a big deal at the time. There were machines that could sequence 384 pieces of DNA at once. They would sequence 1,000 of these letters, if you will, in a row. And again, put together the order. And they do this many times and put together, stitch together the whole human genome sequence. It took about 15 years or so to ultimately do this from technology starting time to final completion. So that's where things stood in 2003. But wouldn't you know it, there was a revolution. And this is what's made this whole field of personalized medicine, at least as we see it getting practiced soon, possible. It's the fact that you can now do this at a much, much more affordable scale and in a throughput that's almost beyond belief. So basically, there are new machines. This happens to be one for Illumina. I forgot to mention I am in conflict here. I consult for Illumina. I'm on the board of a company called Genapsis and a genome interpretation company called Personalis. I forgot to put my conflicts up there, so you should assume that everything I'm telling you is biased, okay? But nonetheless, I'll give you my perspective, and you can take it. My understanding is they're going to put these talks online, so you can filter however you like later if you like. All right, so anyway, these new machines are amazing. They will sequence in one run 2.5 billion pieces of DNA. Now, they'll do it about 200 bases at a time, so 200 of these letters at once is what they'll do. They're not super fast, but they can still do it. And the machines take about 2 weeks to run, and you have to, it's a massive amount of data. You have to offload the data and start analyzing it. And you can do about 20 genomes in one of these machines in about 3 weeks, okay? The cost, these days, we actually pay $3,000 to get a human genome sequence, a research-grade genome, if you will. I'll come back to what that means in a minute. It's not as accurate as a clinical-grade genome, as we call them. And these machines aren't cheap. They cost $800,000 a piece to be able to do this. And so actually getting a genome sequence, getting the sequence is fairly cheap. Actually getting it interpreted is actually quite expensive. It's probably on the order of 15 to 20,000. It takes 200 person hours for us to clinically annotate a genome, if you will. But the point is you can actually do this fairly inexpensively. And so this is all possible because a revolution in DNA sequence started, and it's about 2004. And new technologies have come out. And basically, it's been plummeting about 10-fold every 15 months or so. Now, it's slowed down a little bit over the last 6 months, but it's still accelerating to the point where many of us think we'll hit in fact, there's a big push to hit the $1,000 genome, and that'll probably happen next year or so. Okay, so this is much, much faster than Moore's Law for those of you who were physics majors or what have you here. So the point is that this is a revolution. It's not stopping. It's going to be cheap. There's some people even talking about sequencing genomes for $100, okay, which would be pretty amazing, right? So what this means, and this is something you might think about as we go through the lecture, it's not, can I get my genome sequence? It's whether you want to get your genome sequence, what would you learn for it? And actually, that's going to be a big theme in today's talk, okay? Now, one way I like to think about this in personal terms is I was just out shopping for hot tubs, and I realized that actually the cost of a hot tub is over twice as much as a human genome sequence. So you and your significant other can get 2 genome sequences for the price of a hot tub. And we can argue about what's more valuable. I think they both probably are quite valuable. All right. So as I say, there's a big push to hit this $1,000 genome. Should happen soon, probably 2013. Because this has gotten fairly affordable, and even before it's as cheap as it is now, quite a few well-known people have got their genome sequenced. So this is the guy who solved the structure of DNA, Jim Watson. Craig Venter is a big player in the genome field, and even Ozzy Osbourne has gotten his genome sequenced. And the interpretation of these would be quite interesting, I'm sure. Okay. So what do you do with the genome sequencing? Well, there's a lot of different projects that are going on now. Certainly one of the biggest projects that's out there is something called 1,000 Genomes Project. And it's a little bit of misnomer because the project is now scaled up to sequence 10,000 genomes from individuals all around the world. And this is an area that was actually pioneered at Stanford by people in the biology department like Mark Feldman and others who basically pioneered the analysis of human migration and such and trying to solve this. And now people are sequencing, as they say, 10,000 people from all over the planet to try and understand human diversity and just how much variation is there in the genome. And there is a lot. It's quite remarkable. Okay. How to interpret that is still up for grabs, and that's a big challenge. The main point of today's talk, though, is to talk about how does the genome impact medicine. And so basically, that's what I'm going to talk about. And I'm not an MD, so this is my disclosure. Anything I tell you should not use for medical interpretation, and this is my disclaimer, don't sue me for anything you're about to learn. All right. These are some of the areas in which genomics is going to impact medicine, and some of it already is happening right now. So understanding and treating disease, especially those that are of genetic origin, is already happening now with genome sequencing. And I'm going to talk about a few examples in just a minute. Pharmacogenomics, so looking at your genome to predict your drug response, either your side effects or dosing of drugs, you can now get from a genome sequence. A big area that's up for grabs is this one. How much can you learn about your genome, what you're disposed to for disease risk and such? And that's a big theme of today's talk. That's where my personal story comes in. And then if there's time, we could talk about some of the issues related in this field about genome sequencing in terms of its implications on insurance and privacy and things like that. Let's start with this first one where genome sequencing is having a huge impact on what we call personalized medicine. One of the biggest areas in this field is actually cancer. Many of you know cancer is a genetic disease, both in terms of its predisposition, it can run in families, and also, whether it runs in families or not, just when you get cancer, it's almost always certainly has a genetic basis. That is to say, there'll be changes in your DNA that cause your cells to grow uncontrollably, okay? And it's estimated these days there's something on the order of 10 to 12 driver mutations, 10 to 12 genetic changes that are actually driving these cells to grow out of control, okay? It's not a single change, it's actually many changes. And by the time your tumors and things get big, in many cases, it's actually fairly heterogeneous. Some parts of the tumor can have one kind of changes and other parts can have other changes. But nonetheless, what's driving this is, in fact, these genetic lesions, if you will. Now, late-stage cancer, and for many cancers, it's almost always diagnosed late stage, is a time, late stage, stage 4, is when cancer is metastasized. And so when cancer is spread, it's almost always lethal, no matter what form of cancer we're talking about. And so there's the first push for genome sequencing and personalized medicine was to sequence patients, late-stage cancer patients, because they're basically desperate. And the hope is that you'll find genetic changes that might be druggable. That's the rationale for the whole field. Okay, and in fact, that turns out to be true in a number of cases. It's not standard of care yet, although I do think this will become standard of care in the future. And just to show you what I'm talking about, this is just literally a cancer we sequenced fairly recently in our lab, esophageal cancer. And this particular cancer has about 20,000 small changes. These are these single nucleotide changes, if you will. There are 15,000 of these, and there's some smaller insertions and deletions. And these are these larger structural variants I mentioned, the larger changes, and there's 300 of these, and they'll be represented by these thick lines, the regions that have amplified. There's some thin lines where things are deleted, in certain areas where things are gone. So here's an amplified region and such in this particular chromosome. So the point of showing you all this is to show that most cancers have many genetic changes. And by looking at these and trying to find those that are recurrent amongst different individuals, you can get a sense of which ones might be drivers, which ones might be passengers, meaning genetic changes that have occurred but are being tagged along because they happen to sit next to a driver mutation. Okay, so what's happened is there are big projects out there now to sequence cancer genomes, and some of this is occurring at Stanford. And the goal is ultimately to sequence on the order of 25,000 cancer genomes from different sorts of cancer. And they're not that far along yet. There's a few thousand that have been done, and they're going after some of the big killers. Pancreatic cancer, when it's diagnosed, it's almost always late stage, and by then the prognosis is quite poor. Ovarian cancer is in the same situation. It's the fourth leading cause of death from cancer in the US, and again, that's often almost always diagnosed late, and so on and so forth. Colon is number one. That can often be diagnosed a bit earlier, and so it's still a huge problem, but I don't remember if the number of deaths is as high as ovarian cancer. But the point is a lot of people are dying of these diseases, and again, if it's caught late, people are in trouble. It's what's been found out by sequencing these different cancers, by looking at the DNA, it's been found that certain biological pathways tend to be altered in these cancer patients, and it varies a bit with the particular person. So no one person, it turns out, has exactly the same genetic changes for any of these cancers, and that was what has been perplexing. Sometimes there's a propensity for certain kinds of changes. You may have heard of BRCA1, which is a familial type of cancer. That does show up in a number of cancers, but even in breast cancer, it's on the order of 5 to 10% of patients have a BRCA1 mutation. But there are changes in the BRCA1 pathway. There are other genes in this pathway that can be altered, even if it's not BRCA1 itself. And this is just an example. I know this is complicated. This is a typical scientist trying to overload you with data. But the point is that this is ovarian cancer, and you'll see various signaling pathways that are changed. It won't be any one of these, but in this pathway, so 67% of patients will have a defect, a genetic change in this pathway, 45% in this. It'll be no one particular component, it'll be different ones in different people. And likewise, here's a pathway that involves this BRCA mutation I mentioned. This actually turns out to be what's called a recombination pathway. And you can see that this pathway is altered in many individuals as well. I've lost the percentages. It's up here somewhere. About 33% of patients will have a change in this pathway. So again, it's not the same in every person, nor is it even the same change in this pathway. But together, this is a major pathway that's changed. So on and so forth. Here's another signaling pathway and another one. And then what's happening is in different cancers, you're seeing a different set of pathways, sometimes overlapping between one cancer and another, okay? And so the big surprise from all this cancer sequencing is that, first of all, every cancer is complex. There are multiple changes, and it varies from person to person. And the other big surprise was that a lot of the genes are shared and the pathways are shared between different types of cancer. As an example, non-small cell lung carcinoma, well known for having a particular growth factor pathway, EGF receptor, amplified or going out of control in that, it turns out that that same pathway can be altered in a number of other kinds of cancers as well. And so what's happening is cancers are now not just getting classified by their origin, meaning colon, ovarian, esophageal. They're actually getting classified by the genes that are changed and the pathways that are changed. Why is that important? Well, if you're one of these patients, if you're altered in this pathway, the PI3 kinase pathway, for many of these pathways, they're actually therapeutic targets of yours. They're drugs that will operate in this pathway. And so what's happening, and there's been a few success cases, you actually treat individuals, if you're altered in this PI3 kinase pathway, there are PI3 kinase inhibitors. And so you will treat those individuals with that drug, and they'll actually respond to that. And the point is that there's no sense treating a patient with PI3 kinase inhibitor if that pathway is not changed. And this is why medicine is changing, because in the old days, to be a certain protocol, you have ovarian cancer. First thing you get is you know, X. And if that doesn't work, you get either cisplatin or Taxol. If that doesn't work, you get Y. You go down this pathway. And most of those drugs are irrelevant, okay? Because if that pathway is not changed in your cancer, there's no sense treating you with this. I can give you a personal example. I had a cousin, fairly young, who got non-small cell lung carcinoma, was treated with a drug. We now know what the target is. We didn't know. It's called Iressa. I don't know if any of you have heard of this. But anyway, he was treated with that. 10% of patients are a responder. They didn't know how to predict those 10%. He was not a responder. He died, you know, I think about 6 months later. But we wasted 6 weeks treating with a drug that had no point. Would never could have worked. These days, we know the genetic lesion that's altered, so you'd actually test that first. You'd never go down that route of therapy. And this is where personalized medicine is moving in cancer. Now, don't get the impression that this is solving all your problems. I mean, what it's doing is suggesting drugs. Some of them are experimental. Some of them are actually approved things on the market that you can use. And usually a drug will help, but almost always patients now are going into remission because there's so many cells are changed. Remember, I mentioned there are lots of driver mutations. And so it's clear that what we're going to need in the future are multiple drugs for every cancer, just like they use for AIDS therapy now, where right now for AIDS, right, you don't use a single drug if you want to control AIDS because the mutations arise and actually get around that. Same will be true for cancer. We'll need multiple drugs, but even one drug is better than none, again, when you're at a late stage case. And so we are doing this at Stanford, as are other institutions, to try and to do this. Now, this is fairly expensive. It's all in a clinical research phase. It's not standard of care, but my prediction is that this will become standard of care in the future. And quite frankly, if I had late stage cancer, the first thing I'd want to do is get my genome sequenced. And I've just opened a floodgate to get a lot of emails there. But anyway, I do think that's where the field is going. It's something you should be thinking about as well. Okay. And this slide just shows that this is a classic case of a drug that targets a very specific set of changed genes, and this was used in several pathways. There's a lot of drugs like this, so this will target one particular pathway. The other area in which genomics is having a huge impact is in what I call solving mystery diseases. So you may or may not be aware of this, but a lot of children are born with problems, and if you see the problem running in the family, meaning the grandfather had it and then the child has it, or a parent had it, or a cousin, there's a good chance it's genetic. If it's running in a family, good chance it's genetic. Okay. So for many of these cases, you can actually, like hypercardiomyopathy or cardiac failure, you can actually go in and look at candidate genes and try and see which genes are altered. And ironically, for something like hypercardiomyopathy, there are 3 tests that are commonly used. They'll wind up identifying, I think it's on the order of about 10 genes in this pathway. Each test costs $3,000 to $4,000, and people can spend up to $10,000 running through these 3 tests to see 10 genes. Well, it turns out that will work half the time because there's a lot of other genes involved in this. And so these days what happens is it's often common if you see a patient, a child who is having problems, and you try the obvious candidates, it doesn't work, people turn to genome sequencing with the hope that that will tell them what's wrong with this. And this, I can tell you, is what's being done a lot, or a fair amount, but it fails much more than it succeeds, at least in our hands, but it's where the field is going. And I'll show you one example that was successful. This was done by a group at Baylor, Richard Gibbs' group, where there was a family that had fraternal twins, dizygotic twins, and they actually had a lot of problems. They were floppy babies, if you will, and they had developmental delays and all kinds of problems. It was actually the mom who figured out, I think when they were about 4 or 5 years old, that they had dystonia, and that was the diagnosis that came up, and if you treated them with dopamine, they actually responded pretty well. So they went from being in a lot of trouble to doing a lot better, but they weren't perfectly cured. So what was done by this Baylor group is they went in and sequenced these kids and also sequenced the parents, and then zoomed in and looked at the change pathways. So you're looking at changes that are in the kids that either, at least where both copies weren't changed in the parent. And they ultimately figured out that the likely culprit was this gene here. It was a mutation in a gene that actually is in a pathway that leads to production of dopamine, which explains why if you give the kids dopamine, they do better. But this doesn't only make dopamine, it makes serotonin. And what they discovered is based on this information is that if they not only gave dopamine but supplemented with a a serotonin precursor. It's actually this one here. The kids, in fact, did a lot better. So this is one of the few cases, probably the only one I know, in which actually getting the genome sequence led to information that suggested a therapy that improved the condition of the kids. But imagine you're a parent and you have a child who has a problem, and we are doing this. We do go in and sequence the family and try and see what's wrong with that child relative to the parents and see if we can figure it out. Often we'll see a short list of candidate genes, if you will. It's rare we see a smoking gun that says, oh, this is the altered gene, but nonetheless we're trying to figure this out because the ultimate goal is that could help the family either in future, even if you can't do anything therapeutically, you could actually help them in family planning for the future. Okay, so this is where the field is anyway. So this is all cutting edge stuff. This brings us to this scenario. Somehow I got a slide ahead of myself here. So the bottom line is, okay, these are disease cases. These are no-brainers to be sequencing genomes to see if we can improve the health of the individual. What about if you're a generally healthy person? Can sequencing a genome help you? And if so, how might it help you? And I actually wanted to turn this around to you. How many of you want to get your genome sequenced? Okay, quite a few here. How many don't? Okay. Oh, wow. Normally it's about 50-50. There's no right or wrong to this, by the way. And as I say, I'll walk you through my personal genome, and you can get a sense of what you might learn or might not learn. How many don't know, by the way, whether they want to get their genome sequenced? Okay. So normally when I give this, by the way, it's 50-50. The exceptions are, I remember at a fundraising dinner in this that was like 90% were in favor of getting their genome sequenced. And when I talked at the Stanford Law School, 90% were in favor of not getting their genome sequenced. It's a very risk-averse group, so, and I'm not kidding either. So it was quite amusing. Normally it's about half and half. And it doesn't matter about the educational background generally. That is to say, when I've given talks in areas where people maybe haven't had as much money and such, it's usually still 50-50. So, and again, you might get a sense of why. All right, so why might you want to get your genome sequenced? Well, you might want to know if you're likely to get certain diseases, this predisposition I mentioned earlier. You might want to know your sensitivity to medications. I guarantee you will learn something about that if you get your genome sequenced. And just curious, this is why a lot of people get their genome sequenced. Why you might not want to get your genome sequenced? Well, you may not want to know if you're likely to get certain diseases, right? Huntington's disease is a disease you can figure out, not typically done by the way we sequence genomes now, but there are tests for Huntington's, and there's nothing you can do about it. So you learn you're at risk for Huntington's, you will a very high probability get it, and a very high probability die of it. And actually, and when people learn this, about 25% commit suicide. So there is a big issue about learning this information, okay? Another reason why people say they don't want to know about sequencing genes, well, they're just too complex. Disease is too complex. You sequence a genome, we'll never figure this stuff out, and all it's going to do is make me worry, okay? And a third area is a lot of people worry about genetic privacy, discrimination, et cetera. These are reasons why you might not want to get your genome sequenced, okay? So I'm going to tell you again about my personal genome, and we're big believers that genome sequencing can be valuable. And again, it should be a choice issue. We can talk about that a little more later. And from my perspective, we wanted to know if genome sequencing for healthy people can be valuable for incorporating information into healthcare and managing medicine, et cetera. And so one way of thinking about this is that, again, in a pictorial fashion, is can your genome sequence be useful somehow with your physician to either predict disease risk, diagnose, monitor, treat disease, and minimally understand disease states. And you wouldn't just do this in a vacuum. You'd use family history. You'd use your genome sequence with your family history. You'd also do blood tests, right? We all go to the doctors. I used to go about every two or 3 years, and they draw blood, and they measure about 20 things in a routine test. If there's something wrong with you, they measure more. But it's a very low number of things. And so all of this would go together and somehow be useful in managing your health care. Now, from a lot of work that we do, we know that 20 things is pathetic in my mind. So what we've been doing is actually a lot of what's called Omics Profiling. And I'll talk more about this in a minute. But we can measure in your blood lots of different things. We can measure all of your RNA in your blood cells. We can measure a lot of your proteins called your proteome. a lot of your metabolites called your metabolome. And so what we want to do, and what we do is we measure 40,000 things in your blood instead of 20, okay? Now, we don't know what the heck we're doing. We're measuring 40,000 things. We don't know which ones are valuable as markers. It's really a research project. And this is the plan. And our view is that getting this together with this should give us a much better way to do this. Okay, that was the idea. Now, I was at Yale for 24 years. I moved to Stanford 3 years ago. And we're one of the few labs that actually works in the genomic space as well as in this space as well. We've been talking about this for some time. And when I came to Stanford, it was time to go ahead and see if we could do this and try this. And so, again, the idea was to put together your genome information with lots of other information. This is, again, your DNA, your methylated genome, for those of you who are aficionados here. And this is your RNA, your proteins. We measure about 5,000 proteins. We measure, this is measuring the abundant ones. We have a specific measure for measuring something called your cytokines. Those are a very important class of molecules. They're very non-abundant in your blood, but we like to measure them because they're important, so we set up a profile for this. We follow 5,300 metabolite peaks, although one limitation is we only know what 1,000 of those peaks are. So we're seeing kind of random peaks in a mass spectrometer, but we can follow, we know 1,000 are, which is pretty good. Mind you, it's still better than 20, but it means we have a long ways to go to know what those other 4,300 are. We also follow autoantibody profiles. You have antibodies floating in your sera, and we believe your immune system is an exquisite monitor of disease states. And so technology I invented at Yale was the ability to put down all of your proteins, or nearly all of your proteins, on a slide and look at your reactivity to sera. And so we can see what autoantibodies you have, if you will, what antibodies you're reacting against. And we think that could be valuable for, again, classifying disease states. And we showed this for ovarian cancer, actually, a few years ago. And lastly, one thing we're just starting, it's not integrated in the first profile, is looking at your microbiome, because you may not realize there are a lot more cells in you than aren't you. They're actually bacteria and microbes in your gut, and in fact, you have about 2 kilograms of these, and it's on the order of 100 times more cells are microbes than you, okay? So, and it's very valuable. It actually makes metabolites and other things. And so we're just starting to measure your so-called microbiome, and we do this in a variety of tissues. Okay, so the idea was to see if we could measure all of these things, and we got started with the first bunch, not the methylome, and together generate what we call a personal omics profile. Bring all these different omes, if you will, your proteins, your transcripts, your metabolomes, all together and measure these in a person. And so when I first showed up at Stanford to launch this project, there's a bit of a rage going on. There was, genome sequencing was just started. There were like 2 genomes that were sequenced. And there was, the rage was actually in the medical school, where there was a group of people that said, our med students and grad students are going to have to learn all this stuff. Let's let them genotype themselves, which means let's let them follow about 1 million markers, not their whole genome, but about 1 million DNA markers. So it's a 23andMe test, if you will. Let's let them follow themselves as part of a class. We'll teach a course and let students do this. And it created a big brouhaha because half the faculty said, this is a great idea. Our students have to know this stuff. Medicine's got, our physicians really have to be on top of this. The other half said, this is terrible. Students are going to learn things that are going to harm themselves. They'll learn their father's not their father. Or they got Huntington's or Parkinson's. And they can't handle this information, and we know what's best. So it was very interesting because it was pretty much the basic scientists were 100% in favor, and the clinicians are 100% against. So it was a very interesting discussion, and I walked right into the middle of this when I showed up at Stanford. So I'm scratching my head, and I knew we wanted to do all this because you can tell I'm a big believer in this stuff. So we decided to launch a study, and to get around all the controversy, we picked someone who would give plenty of material and who I knew wouldn't sue me. And that person, of course, was me, okay? So we decided to analyze me, and this is not unusual in the scientific profession. People start with themselves. In many respects, it's a very safe way to get started. Now, when I first showed up at Stanford, I had a role in a health plan, and like any scientist, they took my blood, and they said, oh, give me an extra tube, which I threw in my backpack and put it in my fridge, which is very valuable. And then the day we launched the study was this day, and it turns out I had a rhinovirus infection, so a common cold. So we took blood that day. We did it 4 days later when I was recovering, and 21 days later. And then we would sample about every 2 months or so when I was healthy. And then basically, I have little kids, so I get sick all the time. So 9 months later, I picked up a nasty respiratory syncytial virus infection. It's another cold. It's less common amongst kids, not so much among adults. And I was out for 2 days, which is very unusual for me. Even when I'm sick, I drag myself in and infect everyone. So, but the bottom line was I did get this nasty cold. And you can see as soon as I started getting the sniffles, we took blood, then the next day, then 2 days later, 2 days. You can see a lot of dense sampling, if you will, during this time. And then we went back to a healthy state. We've now been doing this first part was about 14 months, but we've been doing this for almost 3 years now. And I've had 4 viral infections, 2 rhinovirus, both relatively mild, this RSV, and I had a very nasty adenovirus infection. So I not only get sick, I know what I get sick with because we measure all this stuff as we go along. And during this time, we're actually pulling my blood and we're profiling all my molecules to see what's going on. Okay. So we start, mind you, this is an omics profile. We start by sequencing the genome. Again, imagine this is you. What can I learn from my genome sequence? I'm going to walk you through my odyssey. Okay. We knew there were some issues with genome sequencing. People just talked about, oh, sequence the genome. Well, we knew it's not as easy as that. We knew there was actually technical challenges associated with it. So in fact, we sequenced my genome with 2 leading technologies, Illumina and Complete Genomics. This one was less common at the time we did this. And it became very clear to us that none of these technologies are perfect. This is fine for what I call research grade sequencing. Nobody cares if you're 10% wrong. I mean, they do care at some level. But you know, it's your genome, when it's a genome you're going to interpret medically, having 11% errors, if you will, is terrible, right? Because it's a lot of differences that are important. So we sequenced, as I say, with these, we found that only 89%, I guess I'm implying here, only 89% of the variant calls we will. I forgot to say, when you sequence a genome, you run off all these bases and you compare it to a reference. We have some of the world's experts on how to do that comparison here. So you compare it to a reference genome and you look for your changes, your variants. That's how genomes are sequenced. So I did come up with, at the end, 3.3 million high confidence variants, meaning they've been validated by 2 methods, a lot of lower confidence ones, a lot of short insertion deletions, and 3,000 of these large changes I mentioned relative to a reference genome. Okay, that's how many changes I have. Okay. A lot of these variants, I'll skip over this quickly, but they're not just willy-nilly in the genome, they're actually sitting in genes. So our genes that we know the best, they're protein coding genes, and code about 2% of the genome. And so 31,000 of my variants are lying in genes that are expressed as RNA, and about 1,800 of these lead to changes in protein. They're called non-synonymous changes. We can actually find these. And one of the interesting results we found is that these are the alleles that are specific to me. These are the alleles specific to the reference genome. It turns out, what do I mean by that? These are, so remember we have a copy of our genes from our father and one from our mother. Well, they actually don't behave the same. So what this is showing is that during my rhinovirus infection, sometimes my father's alleles are higher expressed and sometimes it's my mother's allele. So in fact, our different genes are behaving differently, and that's why we're going to all be different as well in our responses, because we have, again, 2 copies of all our genes are not going to behave the same. So they will behave differently. So what you get from your parents will probably be critical for what happens to you. All right. So we get this list of variants. It's a huge list, right? 3.5 million high confidence variants. That is things we're sure are a problem, things that are our changes. There's this huge list of changes. Which ones are going to be impacting my health? So we have a process we go through for interpreting this, and this was actually first set up by Ewan Ashley's group with Tul Butte and Russ Altman. We have, at Stanford, we have the premier leaders in interpreting genomes, and this is where I'm conflicted. I confess I'm part of this group, and we founded a company in this area. But the idea is that we go from this list of variants and try and interpret genomes, and even though we found a company, it's still a big challenge, okay, to do this. It's not trivial. And the way you do this is you take all your list of variants, your 3.5 million, you look for those that aren't so common, because the idea, if it was a common variant, I'd probably know about, and it was causing a disease, I'd probably know about it already. That isn't a perfect assumption, but it's not an unreasonable one. We spend most of our time looking at these, the variants that lie inside the genes we know about. And even though that's probably a small fraction of the genes that cause problems, it's the ones we spend the most time on because it's looking under the lamppost. It's the only ones we know how to interpret. So we go into the genome and we look at those that lie inside genes we know how to interpret, those that cause changes in protein. Sorry, this gets a little jargony. Not only do they cause changes in protein, they're predicted to actually screw up the protein and kill it, if you will, damage it. Okay? And then lastly, we look at those that lie inside a gene that's been associated with a human disease, and that's what this database is. So if you don't follow all this mumbo jumbo, the only thing you really have to remember is we go from this list of 3.5 million variants to finding those that are thought to be killing proteins, if you will, that are lying in diseased genes. That's the pipeline. All kinds of assumptions are made. All of them are wrong, but nonetheless, it's a first pass. This is early days in this business. Okay. So at the end of the day, I have 51 mutations in genes associated with human disease, and this is my list. There it is. So here's lesson number 1. If you're a worrier, do not get your genome sequenced. Okay? You will have a list just like this. Okay? It may not be the same genes, but it'll be in the same number. We've sequenced a number of genomes, and they're like this. So what does all this mean? Well, most of this stuff is, believe it or not, is fairly benign, or if it were truly damaging, causing problems, I would not be giving today's talk. Okay? And here's one, polycystic kidney disease. If that were a damaging mutation, yeah, I wouldn't be here. All right? So this is some of the complexities of interpreting genomes. Nonetheless, we did pore over all this. One thing that was refreshing, I knew I was heterozygous, meaning I had one chain in a gene for something called anti-alpha trypsin inhibitor. And actually, we did pick that up here. So that was a nice positive control. I found that very serendipitously, by the way, from many years ago. But it was nice to see it in my sequence. Okay, we also didn't just keep our eye on the rare things. We did look at some of the things that were a little more common. Okay. These are the things that turn out to be interesting from a medical standpoint. When we looked at this is a change in a gene that encodes, it's called telomerase. It adds on DNA onto the ends of your chromosomes. It turns out I have the exact same mutation as people with aplastic anemia. And if you believe the literature, I should have aplastic anemia. So we measured the ends of my chromosomes, and they're just a little bit shorter for my age group. Your telomeres actually get shorter with age, okay? And mine are just a little bit shorter for my age group. Yeah, and the number of cells I have short telomeres, just a little bit more, but nothing to be overly worried about. Also, we went and sequenced my mom because I wanted to assign all my variants to my mom's chromosomes. We wanted to interpret hers. She's 84 years old, does not have this disease, aplastic anemia, and she's going strong, and so everything seems fine. So with that in mind, I'm not terribly worried. So what does this mean? It means some of these changes are actually contextual. So in my background, they're not causing problems, but in, for the people who did the study before, in those backgrounds, they are. And this is some of the complexities about interpreting genomes. Okay? Now, mind you, I thought I was in the clear, and then I saw someone give a talk, and this shows the dynamics of science and how, it's always a moving target. He gave a talk, and they had done thousands of people, and they looked at the ends of their chromosomes. And for women, they have a very slow decrease in telomere length as a function of age. Well, it turns out for men, they hockey stick down at about age 50. So their telomeres get a lot shorter. So even though my mom's in the clear, I'm not necessarily in the clear. I forgot to say I don't have aplastic anemia. That was probably obvious. So in fact, I may not be in the clear. I was planning on following my ends of my chromosomes the rest of my life anyway, but now I have a good reason to do that because it's possible mine will actually drop more dramatically. So this is the kind of thing I could look out for using my genome sequences information. Now, I have high cholesterol. I knew that already long before the study, but I can actually find the changes that are responsible for that. I have two changes, one of my father's. They're not the same change. One of my father's chromosome and one of my mother's in this gene, and that's likely responsible for this. And I also had some changes which were curious to me at the time that were suggested as higher risk for diabetes. These aren't definitive changes, but they put me at increased risk. Now, Atul Butte's lab is an expert at actually integrating over lots of genes, if you will. So we have what are called risk alleles, changes that increase your probability of getting a disease, several fold, if you will. And what you can do is you can actually sum over all these. There's been lots and lots of genetic studies over the years for lots of different diseases. And here's a list of some of them. Actually, the list goes way off the floor when you look at the whole list. But these are the diseases, and you can actually integrate over these various changes and see by adding them up, summing them up, if you want a complex program, you can see whether you're at increased risk or decreased risk for certain diseases. Okay, this is what 23andMe does when they actually look at 1 million changes in your genome. We're looking at 6 billion, okay? So we have a much better way of doing this. And so when we did this, we looked at this, and this is as a European male, so I'm of European descent. That triangle is my starting point, if you will. There's a 100% chance of getting this disease at this line. This is about a 0% over here. So triangle is where my starting point is. And if I'm at increased risk, I have this yellow-orangish line to the right. And if I'm at decreased risk, I have bluish line to the left. And the bottom line is a lot of this made sense to me already. I'm at increased risk for coronary artery disease. I knew that. Everyone on my father's side has died of heart failure. So that one made sense. Here's my high cholesterol and high lipid problem right here, increased risk, and so on. And then low obesity, that's another one that's up here somewhere. That one I knew about already as well. Nobody in my family is overweight. But there were some big surprises. This one's fairly new, increased risk of basal cell carcinoma. And I found out recently, we had a family reunion in August. I was not aware of this in my family, but it turns out both my brothers have had a number of these removed actually over the years. So in fact, it was in my family, I just didn't know. We don't talk about this around the dinner table, right? It never came up. And so I wouldn't know my father's passed away. I don't have that information. Here was the one that popped up very early in the study, which was at an increased risk for type 2 diabetes. And that one came out of the blue as well. I was not aware of that running in my family. So because we had been doing all these other measurements, remember I mentioned metabolites and protein and RNA, we've been following all these things, including my glucose levels. So diabetes means increased glucose levels. And my levels have been running along just fine. This is in a fairly normal range, whether it's fasting or not. But because my genome said I'm at risk for diabetes, I was very perplexed. I went to the world's experts here, Jerry Reeven's group, and asked them you know, if they could do, they have a glucose metabolism test here. And so, you know, that took 5 months between my busy schedule, and it's a very high demand clinic there. And so they basically agreed and set this up. And the day I went into that was actually this day here. And I should say when I first walked in, you know, I was consulting with the woman running the test, and we both agreed it was incredibly unlikely I would have diabetes because, you know, I don't fit the normal stereotype for a diabetic. They're typically overweight, and I didn't have a family history. But we did the test anyway. And this is just after fasting. It wasn't even the glucose test. It came out high. 127 is actually not normal after fasting. That's high. And we both perplexed. Repeated it, same number. So that prompted me to do more frequent sampling. Looked again a week later, and once you know it, it shot up. And there's a better test for high glucose, so it's called hemoglobin A1C. And I was 6.4%, which is high, actually, quite high. So I didn't know anything about diabetes. It wasn't running in my family. I never really paid attention to it. It's not my area of research. So I went to my general practitioner, and you know, again, I'm busy, so it took several weeks to get there. And that was one of these days here. And once you know, my glucose shot up even higher. Okay, and well, I should say, when she looked at me, she said, "There's no way you have diabetes. Those guys get the test so wrong." You know, she repeats everything. She probably sent them to the exact same place where the other tests were done. But nonetheless, it came out a lot higher. And you can see my hemoglobin A1C was even higher. 6.7%. And at that point, you know, it's classified as diabetic. 6.5 is the threshold. So with that, I can tell you what day that was. I got back the results. That was the day of the measurement. Day of the results was April 13th, because on that day, I completely changed my lifestyle. So I was the world's worst eater. I ate lots of desserts, ice cream all the time, because I had no reason to think I was going to get diabetes. So I did exercise a certain amount. I ride my bike to work quite frequently. But when I got this news, I doubled my biking. You know, no more excuses on a little drizzly day to not ride my bike. I'd ride my bike, and I ride my bike downtown, which I did this morning for a dentist appointment. And so I doubled my biking. I started running, and I don't have the whole picture here, but it took about 6 months to bring this down to normal. So what was the interpretation of all this? Well, first of all, my genome, in fact, did predict as a risk for a disease. I had no idea it was in my family history. And in fact, by careful monitoring, I saw I did get that disease. And the nice thing was it was a pretty manageable disease. I could change it. I didn't have to take any drugs, thank goodness. But I was actually able to bring it under control. And so now I'm classified as managed. I'm still diabetic, but classified as managed. Okay, so why did all this happen? Well, the interpretation is my genome has me predisposed to diabetes, but that by itself didn't do it. You'll notice when this shot off the roof, it basically came right after this nasty RSV infection. That nasty viral infection. And there have been links between viruses and type 1 diabetes, stress responses in type 2 diabetes, but to our knowledge, there have been nothing published, and still haven't found anything about viruses triggering type 2 diabetes. And so our interpretation is that my genome has me predisposed, and the viral stress response triggered the disease. That's the interpretation. It certainly sets up some obvious hypotheses we want to test. How often does this happen? We published this in March. I can't tell you how many stories I've gotten with people. It's all anecdotal. People in very, very similar situations who've said, you know, we found the exact same thing, that this, after they had some nasty infection, they got diabetes. And so, in fact, it may be more common than people realize. Again, we need a nice systematic study. By studying me in detail, it's led to some obvious hypotheses that we should be pursuing. The other thing I'll tell you, it's getting a little later, so I'll have to wrap this up. Sorry, I lost track of time here. But when you sequence your genome, you can figure out what drugs you're either more risk for side effects or figure out dosing. So, for example, I am on statins. I'm a great responder. But I know which statins I shouldn't take that could cause some nasty side effects. And so I can get that from my genome sequence. I also see, I hope I never need any of these things, but I see an increased sensitivity for warfarin, which is used after heart attacks and so on. And interestingly, there's 2 diabetic drugs. This is a frontline drug for diabetes, metformin. Okay, I know that I would have an increased effect, so I should start off with a lower dose based on my genome sequence. So it can, if nothing else, you'll learn something about your drug dosing and responsiveness from your genome. Okay. So because we've run out of time, I can tell you we've done lots and lots of other studies of all my other omes to follow all the different molecular pathways that were changing while I got sick during these viral infections and as I got diabetes. It was quite interesting. From a scientific standpoint, it's really fascinating. So scientifically, there's a very interesting project. Personally, it wasn't perhaps the outcome I was hoping for in this whole, when I entered this study. It was really a proof of principle study that turned out to be quite fascinating. And I can tell you, imagine you're studying 40,000 data points across, we've now done 44 samples. I just did one last week. So it's really a complex data set. It's very diverse data type. So again, we're in the heartland of where you know, computer science manifests itself extremely well. And so the bottom line is we've come up with, we think, some very field-changing computational frameworks for being able to analyze these sorts of data. There are 30 terabytes, for those of you who follow this sort of thing, around my genome just from the first half of the study. So we're probably getting, there's even more later, so we're probably getting up close to 100 terabytes by the time this is all done for the study. So it's going to be a lot of information just on one person. Okay. So the bottom line is we can follow all these different pathways that are changing. I won't have time to do this, but this is during the RSV infection. These are all the components, metabolites, proteins, and RNA that go down, the ones that get greener, if you will. And these are the ones that go up. And you can see the pathways that change. There's something called secretory pathway. And you can see my insulin pathway has actually changed. It actually went down during this time, between the time I got diabetes. You can see some other pathways spiking up, a glucose sensing pathway that just went off the roof, and an insulin response pathway that went down through some spike time. So we can see in molecular detail what's going on. We don't know which of these 40,000 are going to be great biomarkers, if you will, for the disease and how this will help us treat the disease. Diabetes, I think, is very well suited to this kind of analysis. You probably know there's about a quarter of people are overweight right now, and it's estimated that as much as half the population will be diabetic by 2050, maybe even more by current estimates. And so obese and diabetic. And so it's going to be astronomical. Our own view is that this is a very heterogeneous, it's not one disease, it's 100 diseases. That's why some people respond to metformin, some don't. That's why it's been very difficult to map genetically. We think the kinds of profiles we're doing here, these omics profiles, will be great for stratifying the disease so we can treat it as 100 diseases. So if we subclassify it, presumably, type X gets this kind of treatment, type Y, that one, type Z, and this is the hypothesis we're pursuing. We want to tear this disease apart and treat it as it should, the heart of personalized medicine. All right, so in my mind, this is really the future of medicine, and again, this is my personal editorial opinion. You can take it for what it's worth, but I'm a believer that people could get their genome sequence at birth and probably should. Doesn't mean that information should be returned to them necessarily at birth. I think it should be up to them. I think your genome belongs to you, and you decide what information you want to get back. Not some physician decides this. You decide what information you get back. I think, nonetheless, some of the obvious things, if there's some very treatable things, your physician could act on that instantly if that's important. And in the New York Times today, or yesterday or today, you may have seen about doing very early genome sequencing for making diagnoses. And in fact, so in a sense, that's happening with that, with some new instruments. I'm a believer that we won't do blood tests of 20 things in the future. I think there'll be many more. I doubt that there'll be all 40,000. I don't know that we need all 40,000 just yet. But we don't know which of those 40,000 we need for predicting what kinds of diseases. And so I'm a believer that we want to get these longitudinal profiles for 1 million people and then use that information to be able to predict disease. Put it all in a giant database and have data-driven medicine. You may have heard this term before, but I really think that's medicine of the future. We have a big database. You have your genome with your variants. You have your electronic health records. And you have all your molecular information and all the treatment information. You see who responds based on what kinds of variables. If you have this variant, you'll respond well to this drug. If you don't, use something else. And you see how people behaved. And imagine a world where there's millions of genome sequences with millions of health records, with millions of treatments. It will completely transform medicine. So I think this is where we're going. So these are the conclusions. By the way, we're not there yet. Long ways to go, but that's where the field is headed. But I do believe personal genome sequencing is here again. Not whether you can get your genome sequenced, or not if you can get your genome, it's whether you want to get your genome sequenced. You guys can decide. I do think it has some value. Is it going to be better 3, 5 years from now? Absolutely. But we can still learn things now that can be valuable. I do believe if we add these other omics, the proteome and other things, we'll get a much clearer idea of what's going on, so we're actually expanding our study. And I think this is a very, very important thing. You can learn a lot from just studying one person. This, I think, surprised a lot of people. People are very used to the idea, well, you have to study 100,000 people to see, compare the patients and control. This is a very different way of thinking. And this is what personalized medicine is all about. I want to understand my altered disease state relative to my healthy state, and I would argue that's true of all of you. I don't care what my altered state looks like relative to all of you healthy people, okay? Because that's not so useful to me. I want to see these changes that occur, catch them early, and treat them, and that's the nature of medicine. Catch it early, treat it, it's often manageable. Catch it late, like cancer, it's often a problem. So I do think this is a big issue. There are lots of issues around concerning costs of doing all this sort of thing and discrimination issues. We can talk about that in answer. I just want to say in my lab, these are some of the spearheads of this work: Rui Chen, George Mias, Hugo Lam, Jennifer Lee Puktan. A large team of people wound up participating in the study, and we had great help from Russ Altman, Atul Butte, you and Ashley, Mark Gerstein, Carrie Nadeau, Hua Tang. And I always like to thank my mom, who helped with the study in many different respects, as you might imagine. So, okay, I think I'll leave it at that. Thanks very much for having me. Yeah, in the back, we'll start there. Is that? Well, 23andMe will do your exome, meaning your coding sequences, which I mentioned. That's actually the part we analyze the most. I don't know what their cost is. You can visit their website. I'm not conflicted with them. So, but I would argue that the interpretation of that is still difficult. This is where Stanford excels. So you can get your genome sequenced these days for not a huge price. I'll bet it's under $10,000 to at least sequence your coding sequence as a layperson. But the interpretation may not be as good. So it takes us, again, 200 person hours to clinically annotate a genome, look for all those telomerase changes of the enzyme that affects the chromosome end. So it's still a big thing. But it will come. There's this whole big issue, too, about direct-to-consumer that is starting to bubble up. Yeah, I think you were next, yeah.
Is the data format standardized so that you get the genome from any of a variety of sources and they'll be able to read each other's files and so on?
Yeah, that's a great question. So the answer is sort of. You can get lists of variant calls, but different algorithms have different biases and ways. So, when we talk about getting this list of variants, but it's not a perfect business. There are a lot of the genome where things look very similar but not identical to one another. And so the different algorithms have different problems with some of those regions. And they'll have different biases. So, and different people use different algorithms. So it's largely, it comes out in the same format that I could compare my list of variants with your list of variants. But if you processed yours in a different way, some of those differences may be due to the algorithms and not the and not reality, if you know what I mean. So that's some of the challenges as well. So typically, when you're doing a study, you go in and you reanalyze all the genomes with a common format so that all the biases will at least be the same. Okay. It's a great question, though. Yeah, I'm not sure who is next. Okay, I'll let you decide. Yeah.
I understand that sperm have been identified as a rapidly changing genetic component, and I guess cancers would be another type of cell that's changing very rapidly. How do you keep track of those? I mean, I understand, my guess is you're sequencing a certain reference cell, but what do you do with these cells that are changing faster than
Well, this is a great question. Just how much is our genome the same across different tissues? And we actually have a paper coming out. I didn't get into details. First, we started by sequencing my blood and my saliva just to see how many differences there are. And there aren't many, actually. We couldn't find many. We do have a paper that's coming out where we did different tissues from an autopsy patient. We're looking, and you do see differences in these structural variants across different tissues. So there are somatic changes that occur. They're very rampant in cancer, especially many forms, because actually they've been designed to mutate a lot. Those cells often are defective in repair pathways, and that leads to all kinds of chromosomal changes. So they're a special breed that just kind of goes ballistic, if you will, in terms of genetic changes. And sperm, I think, less so. But so there are somatic changes that occur, and some of those somatic changes probably led to the start of cancer in the first place for many people, almost certainly. Yeah.
Your prediction that the cost of doing the sequencing is going to come down to $1,000 or whatnot, $100. Is it based? This is coming from an industrial engineer, okay? Mm-hmm. So is it based on manufacturing cost? Are you assuming that there's going to be thousands of these machines built, or are you also assuming that there's going to be an increase in the effectiveness with which you do the sequencing itself?
Well, I think there will be thousands. There already are thousands of machines. There'll be a lot more. There'll be more benchtop things. PCR is a good example, if you've ever seen, for those of you who might know this, where you can amplify DNA. That used to be a special machine. Now there's one on every lab bench. I could envision the same thing happening with sequencers as they get cheaper. There's a new breed of instrument that's coming out that claims the list price will be somewhere on the order of $230,000, so a fourfold drop in price. Imagine that keeps plummeting, and I've heard people talk about cheaper instruments. And then they will become affordable. There might be one in the doctor's office. A big challenge, though, is this accuracy issue that I mentioned earlier. For the genomes we care about, we actually use two technologies. I forgot to mention that, to get a highly accurate sequence. Because, again, you don't want to have 10% errors in your genes, because that leads to all kinds of missed calls and things that you want to watch out for. So I do think we need improvement in the accuracy. I do think there'll be a plethora of these machines everywhere. And I could see it becoming $100. There are a lot of technical reasons why it'll drop. Right now we do fluorescence-based methods, and that is kind of expensive. These fluorescent dyes and special nucleotides are expensive, but electrical readouts are going to be a lot cheaper, and that's how some companies are moving. And there's other interesting designs that are out there. I don't know who's going to win in the long run. I know who's winning now, and which kind of design will win.
So let's hope that you're right, that it'll be $100.
Yeah.
I'm curious about the 200 hours that it takes to analyze all of this. The cost isn't factored into that $3,500, is
That's
it?
right.
Oh, it is, in fact.
It's not. No, it's
not. It is not.
That's right. You're right. It's not factored in.
So getting that information without that, for me, would be
That's right.
almost useless. So about how much would that cost?
Well, right now, I don't know the number. It's probably 15. I may be wrong, but not more than a factor of two, probably. But it's probably about $15,000 in terms of manual costs to go through this. And there's a whole. We have genetic counselors talk to people if any information gets returned. It always gets, by the way, validated in a it's called a CLIA test before it would get returned to a patient.
So how do you go about finding these people? We could come to you and...
Well, we're not set up as a commodity for this. Again, it's a research endeavor, but we do, it depends, you know, many of you, I don't know, a number of you probably are associated with Stanford here, maybe have a doctor here, and you could try and work through that angle, I would say, for starters. I guess if you need further information, contact me. You know, realistically, we can't sequence everybody's genome in this room and interpret their genome. You would just shut down my whole lab. So this is a challenge. It's a huge
One
challenge.
other little sidelight is you mentioned that people can request not knowing certain things.
Well,
But
I
if
think
you
that's
find
how
some-
it should proceed, yeah.
Yeah, but if you find something that you think is highly significant to the health of that person, would you at least say you might want to know more? Or
This is, yeah, I think you should give people the option. You know, this is being heavily debated. By the way, back on the genome sequence, it will get faster and cheaper quicker because we just need to get algorithms set up for this. We're just clunky at this right now, but it will get cheaper, and it's just lagging. Before with sequencing, now it's interpreting genomes, and that we'll get more robust at that. I think it'll drop less than 1,000 at some point for that. On the issue about returning information, it's a great question. You know, what should people learn? Who's in charge of that information? Give you lots of examples. Everyone agrees that learning you have a terminal disease like Huntington's should be up to the person. Nobody argues with that. What about Parkinson's? Well, most people say that's probably up to the person as well. All right, what about breast cancer? BRCA gene running in a family, good chance, you know, one of the family members has it. It's up to them to decide if they want to get that sequenced or not. Suppose it got sequenced for some other reason, they had cancer. You discover they did have the BRCA mutation. Do you tell the kids? Do the kids have the option? No. These are heavily debated questions. I would argue it's up to you want to, that's why you have a genetic counselor, I would call them a genomicist, who actually talks to the person, gets a sense of what information they want returned to them, and then they get to decide. That's how I would manage this. And it's tough to say without giving the answer, you know, what is it you want to learn, what is it you don't want to, but that's where you need a dialogue, and that's why you have a physician and your genetic counselor / genomicist who work that out and return information to the best possible means, I guess.
We're about 5 minutes over, so I think
Yeah.
we have time for one more question, and this gentleman back here has been waiting very patiently.
Yeah. I can hang around afterwards, too, if any of you want.
Thank you. As a result of the infection that you had a change? Was that a permanent change? Would you need to have your personal test again?
Yeah, this is a great question. It's unlikely my genome has changed much, but whether my epigenome, the methylation pattern in my blood cells and things has changed, it is a possibility. Probably not so much from the infection, but probably the fact that I've completely changed my lifestyle. I haven't eaten any sweets or desserts since April 13th of 2011, except for one bite of wedding cake, which frankly tasted terrible. So I'm not missing anything there. So I don't know. You know, I may have changes of that sort, but it's unlikely there have been large changes in my DNA just because that doesn't happen rapidly, unless there were a specific change that stimulated that to happen. So it's a good question, though.
Are you still drinking wine?
Yeah, it turns out wine doesn't have a lot of sugar, so I'm happy to report. Okay. So thanks again for having me. Hi.
Who down in San Diego is sort of doing the same kind of research you are?
Well, from the DNA sequencing side, there's a group at TGen, Translational Genomics Institute. If you want, yeah, they're sequencing genome, but typically only for diseased people.